Hello everyone, welcome back once again to this educational channel called Learn Bio with Janet. Today we'll continue to discuss Form 4 Biology, Chapter 11 of the KSSM syllabus, entitled Immunity in Humans. Subtopics involved here are 11.1 and 11.2. So we're going to study for 11.1, the third line of defense. In the previous video, we've already discussed the first and second line of defense. So in this video, we're studying the third line of defense that involves the lymphocytes and the production of antibodies to destroy antigens. Then for subtopic 11.2, we'll be discussing the action of antibodies. So we're going to discuss this fascinating white blood cell called the lymphocytes, which are like the body's elite soldiers. So I call them the superheroes of the body because they defend the body against antigens, such as pathogens that have entered the body and passed the first and second line of defense. So the lymphocytes are very specialized cells that can produce specific antibodies to destroy the specific antigens. Let us find out more about this fascinating cell. The learning outcomes for today's lesson are as follows. For 11.1, body defense, after the lesson, you should be able to define immunity, antigen, and antibody, as what we have studied in the first video, and then be able to describe the third line of body defense in humans, which includes the antibodies and memory cells and lymphocytes. For 11.2, subtopic 11.2, actions of antibodies, after the lesson, you should be able to discuss the actions of antibodies on foreign antigens, and the actions include neutralization, agglutination, precipitation, opsonization, and lysis. Here's an overview of the body's defense mechanism again, as was shown in the previous video. So for the first line of defense, which is to prevent the entry of pathogens and antigens, we have the skin, mucous membrane, lysozymes, blood clotting, and hydrochloric acid. For the second line of defense, which we have also discussed, we have discussed it in the first video. This includes fever, phagocytosis, and inflammation. So today we are going on to the third line of defense, which is actually the immune response carried out by lymphocytes, which produce antibodies to destroy antigens that have entered the body. Let us first recall some of the terms that we have learned in the previous video. A pathogen is a microorganism that causes disease, such as bacteria, viruses, fungi, and parasites. An antigen is a foreign particle that enters the body and stimulates the immune response. That means the production of antibodies. Okay, so any foreign particle that enters the body and stimulates the immune response is considered an antigen to the body. So these two substances, pathogens and antigens with the GN, are the substances that are harmful to us. All right, cells or substances that are harmful to us. So an antigen can be a toxin, or it can be a protein molecule on the surface of the invading pathogen, such as pathogen here, which can be a bacteria or virus, and then on its surface there may be protein molecules. And so when the lymphocytes detect these pathogens, they will recognize these as not from the body. These antigens are not from the body. So they are recognized as foreign particles, and thus they need to be destroyed or eliminated. All right? So the pathogens carry protein molecules on their surface and other types of molecules that can be detected as foreign particles by the lymphocytes, our white blood cells. Then the antigens will stimulate the lymphocytes to produce antibodies into the blood flow in order to destroy these antigens. So when we think of antigen, we can think of it as an antibody generator. Now this antibody is not generated by the antigen, but it causes the lymphocytes to generate or produce the antibodies to destroy the antigens. So basically, antigens stimulate lymphocytes to produce antibodies to destroy them. All right, as we can see here, lymphocytes produce antibodies which have a Y shape, and they will combine with the antigens, and after that they will destroy them in various ways. So an antibody is a protein found on the lymphocyte surface or a protein produced by lymphocytes into the blood plasma to destroy a specific antigen, such as agglutinin. All right, so this is the good guy, the antibody. And why do we call it an antibody? Well, think of it as a substance that acts against. Anti means lawan or acts against. Body. Now, it's not your own body, but this is the body of the bacteria or the pathogens or the parasites. So it acts against the cells of the bacteria or virus and other pathogens. Okay. Now, the main cell that we're going to discuss today, the star of this video, is the lymphocyte. So nowadays we have many fictional stories about superheroes, but do you know there is a real life superhero in our body, the superhero of all the cells, and these are the lymphocytes, which are leukocytes or white blood cells involved in the immune response to produce antibodies. So this is the main defender of the body. And when the antigens have entered into the body, the lymphocytes are able to recognize these antigens or foreign particles, the bad guys, and after that they will produce specific antibodies to destroy the antigens. So the antigens are located on the surface of the cells, such as the pathogens, right? So lymphocytes are found in the lymph nodes and blood, and if you cut through them, through a cross-section of the cell, you will find that there's a big round nucleus in the center, and the cell is more or less round in appearance, okay, with certain projections sticking out from the cell surface. So here are some notes about what we have discussed earlier. Let me read through it very quickly. The third line of defense. If pathogens overcome the second line of defense, the third line of defense will be activated. It is specific in action because each antibody can only act on a particular antigen. All right. So unlike the first two lines of defense, which are non-specific, that means they can act on all types of microorganisms, the third line of defense is not so. It is specific in action. Now, the third line of defense is the immune system or the immune response, which involves lymphocytes, the elite soldiers in our body, which produce antibodies to destroy antigens. Okay, remember this definition of the third line of defense. It involves the immune system, lymphocytes, produce antibodies to destroy antigens. Definition of an antigen: a foreign particle that enters the body and stimulates the production of antibodies. Antibody: a protein produced by lymphocytes in response to the entry of an antigen into the body. An antibody helps to destroy a specific antigen. For example, lysine causes the rupture of bacteria. Now, characteristic of an antibody. The action of an antibody is specific. That's why we say that the third line of defense is specific. Now, what's the meaning of the word specific? It's quite similar to the characteristic of an enzyme, which is also specific, right? One enzyme can only act on one substrate, one specific substrate. So same with the antibody. Each type of antibody can only combine with a certain type of antigen to promote the destruction of the antigen. That's why we say the third line of defense is specific. Let me give you an example. Let's say you have the antibody for chickenpox. Now, it can only act on the chickenpox virus and destroy it, but it cannot destroy the measles virus or the flu virus. Okay, that's the meaning of the action of an antibody is specific. Now, a question that can be asked in the exam. Sometimes a person may experience swelling of the lymph nodes, especially those in the neck region, all right? Or sometimes the tonsils, which are also masses of tissue that are connected to the lymphatic system, that may also swell. So what is the reason for the swelling of the lymph nodes? So it shows sometimes that there's an infection in the person where pathogens have entered the body. So when a person is infected with pathogens, a lot of lymphocytes will accumulate in the lymph nodes. The soldiers, our body soldiers, the lymphocytes, will be deployed. They will be transported there. They will accumulate in the lymph nodes, and they will quickly attack the pathogens by producing antibodies in order to destroy the antigens or foreign particles, which could be the pathogens, and thus the lymph nodes will swell. The fluid will accumulate there with the cells, all right? Furthermore, the lymph nodes also contain macrophages to destroy the bacteria, dead tissues, and foreign particles through phagocytosis. All right, so there are two modes of defense that are carried out by the lymph nodes. The first one is the lymphocytes inside the lymph nodes will produce antibodies to destroy the antigens. And secondly, the phagocytes, such as the macrophages, will destroy the antigens through phagocytosis, which is by engulfing them and then destroying them or digesting them using enzymes. Right, let's continue by discussing the lymphocytes. So actually our body has a few different types of lymphocytes, but the two main types are the T lymphocytes and the B lymphocytes. Okay, so T lymphocytes attack cells infected by pathogens. Now they all have the same structure, a big round nucleus in the center. So the T lymphocyte attacks cells infected by pathogens. Cells that are already infected by the pathogens have the virus or bacteria inside them. The T lymphocyte will quickly attack these cells to eliminate them so that the pathogens will also die in the process. And they also coordinate the immune response, which is the production of antibodies. So which cell produces the antibodies? It's the B lymphocytes, which is also called the plasma cells. Which are also called the plasma cells. Now these produce antibodies to destroy antigens. Now think of the word antibody, B here. So the B lymphocytes are the ones that produce the antibodies, not the T lymphocytes. So B lymphocytes produce antibodies to destroy antigens. And it's the T lymphocytes that stimulate the B lymphocytes to produce memory cells, which are special lymphocytes that can sort of remember how to produce the same antibody again later on in the future in case there is another attack by the same antigen. So they are called memory cells because they sort of remember how to produce these same antibodies to destroy the same antigens should there be a second attack in the future or a third attack. Here are some types of T lymphocytes and B lymphocytes that we need to know for this syllabus. It is not a complete list. Now, cytotoxic T cells. So T lymphocytes, some of them are cytotoxic T cells that attack and kill the cells already infected by pathogens so that these pathogens cannot reproduce in them. All right? And also, they kill cancer cells to prevent cancer in the body. Then we have the helper T cells, and these are very important because they are the ones that coordinate the immune response when there is an antigen in the body. So they will stimulate the B lymphocytes to divide, become more in number in order to produce more antibodies. They will stimulate the B lymphocytes to produce the antibodies, specific antibodies against that particular antigen. And they also stimulate B lymphocytes to produce memory cells that can last for years. Okay, so we discuss the memory cells further down here. Now, B lymphocytes. Okay, these are two types of B lymphocytes, the plasma cells and the memory B cells. Plasma cells are the actual cells that produce the antibodies to destroy the antigens. Okay, they are a type of B lymphocyte. So in the syllabus, the name is not, the specific name, plasma cell is not stated. So it's up to you whether you want to remember it or not. You can just call it a B lymphocyte, and the function is to produce antibodies to destroy antigens. So T lymphocytes don't produce the antibodies. They just stimulate the B lymphocytes to produce the antibodies. Now, memory B cells or memory cells are the lymphocytes that live for years in the blood. So after the first attack by the pathogen in the body, And after the person has recovered from the disease, such as chickenpox, the lymphocytes have already produced the specific antibody to destroy the chickenpox virus, right? So some of these lymphocytes live on for years in the blood, and they provide the immune system with a long-lasting memory of that specific antigen. The long-lasting memory for, let's say, chickenpox virus, and also they are able to produce the same antibody to help the immune system to respond faster to future attacks. So in case the person is exposed again to the chickenpox virus for the second time or third time, his lymphocytes, the memory cells, will be able to respond by producing antibodies more rapidly than before, and also in at greater levels. More antibodies, and also producing the antibodies more rapidly than before. Now let us discuss this question. What are memory cells? So this can be a structured or essay question. Let's take the case of a person who falls sick because of infection by chickenpox virus. All right. So when the person is sick, his lymphocytes will start to produce the specific antibody for that disease, for chickenpox. And these lymphocytes produce the antibody, specific antibody against the chickenpox virus in order to destroy the virus. So when he recovers from the disease like chickenpox, the person already has some lymphocytes that has produced the specific antibody for chickenpox virus. And these lymphocytes, some of them will remain for many years in the body as memory cells. They do not die, right? So we call them memory cells or memory lymphocytes because they are able to produce that same specific antibody for the pathogens of that particular disease. Again, should there be future infection by the same type of pathogen or antigen. Okay, so they seem to have what we call the memory of how to produce that specific antibody. All right. Therefore, these memory lymphocytes will help defend the body against future infection by the same antigen, and they will start a more rapid immune response if the body is again infected by the same virus or bacteria or antigen. So if the body is infected a second time or a third time and so forth, this time the immune response is faster and more antibodies are produced. Greater numbers of the specific antibody is produced, and at a higher or faster rate, all right? So in that way, the antigen or the virus or bacteria will be destroyed even before it can cause the person to fall sick. So the person will not fall sick. And we say that the body has become immune to the particular disease. That means even when the person is exposed to the virus or bacteria for that particular disease, let's say the person is exposed to the virus that causes chickenpox, he will not fall sick because his body is able to produce large amounts of the specific antibody to destroy the pathogen before it can cause any harm to the body. Now, number 9, lymphocytes are produced in the bone marrow. So after that, some of them will migrate to the thymus, where they develop into T lymphocytes. This is how the T lymphocytes get their name, from the word thymus, right? Now, there are other lymphocytes that still remain in the bone marrow, where they will develop into B lymphocytes, B for bone marrow. Okay, so T lymphocytes, as we have discussed just now, will help to destroy the cells that are already infected with the pathogens, and they also destroy cancer cells. They do not produce antibodies, but whereas the B lymphocytes are the ones that produce the antibodies. Okay. Now, lymphocytes are found in high concentration or large numbers in the lymph nodes. Let us go on to discuss subtopic 11.2, the actions of antibodies. So there are a few different types of antibodies with different actions, but the basic mode of action of the antibodies are the same. Firstly, the antibody will bind to the antigen to form an antigen-antibody complex. So the antigen-antibody complex is just a particle that consists of the antibody molecule and the antigen that are bound together. Next, the antibody acts on the antigen through neutralization, opsonization, lysis, precipitation, or agglutination. These are all the different actions of the antibodies. Lastly, the antigen, such as a pathogen, is ingested by the phagocytes through the process of phagocytosis, where the phagocytes ingest and then digest the antigens to destroy them. So in that way, the phagocytes also clear up the mess. Let us discuss the general mode of action of antibodies. So here we have a lymphocyte in the bloodstream, and it has antibodies, which are proteins found on the surface of the lymphocyte, or these antibodies can be released by the lymphocytes into the bloodstream, into the blood plasma, when there is an antigen, right? So when the lymphocyte recognizes an antigen, which are foreign substances found on the surface of the pathogens, for example, then it will produce the antibodies, which are Y-shaped, and they are made up of a few polypeptide chains. Okay, each antibody is a protein molecule that is made up of a few polypeptide chains. Now, at the ends of this antibody molecule, there is an antigen receptor site. Okay, so it's a yellow colored part here and here. So for the antibody to act on a particular antigen that has invaded the body and passed the first two lines of defense, the antibody must have a complementary shape to the antigen. They must have matching shapes. For example, this antigen, the shape of this antigen matches the shape of the antibody here. Okay, at the antigen receptor site. Thus, this antigen can bind to the antibody. Okay, so the antigen molecule combines with the antigen receptor site found on the antibody molecule to form the antigen-antibody complex, which is a particle consisting of the antigen and the antibody that are bound together. Now, after that, the antibody can destroy the antigen, and there are various ways that the destruction of antigen occurs. Okay, after that, phagocytes will move in to ingest the particles. So the antibody must be complementary in shape to the antigen. Thus, each type of antibody can only combine with a certain type of antigen that has the matching shape to the antigen receptor site on the antibody molecule. So these two antigens here do not match. Their shape is not complementary to that of this antibody molecule. Thus, they cannot bind to the antibody molecule, and there is no destruction of these two antigens. Okay, so we say that the action of the antibody is specific because each type of antibody can only combine or bind to a certain type of antigen, which is complementary in shape to the antibody. All right. Now, lastly, this reminds us of the lock and key hypothesis. All right, the shape of the enzyme, for example, the shape of the substrate must match the shape of the active site on the enzyme. Okay, So this is a similar example here, where it's something like the lock and key hypothesis, where the shape of the antibody must be complementary to the shape of the antigen before the action of the antibody can occur. Now we're ready to discuss the various actions of the antibodies. So antibodies destroy antigens through several mechanisms, such as neutralization, which is carried out by the antibody called antitoxin. Opsonization is carried out by the antibody called opsonin. Precipitation is carried out by the antibody called precipitin. Agglutination carried out by the antibody called agglutinin. And lysis is carried out by the antibody called lysine. Now, to remember these 5 mechanisms of actions of antibodies, remember this memory tip. No PAL. So think of the antigens like bacteria or viruses that have entered the body. They have no PALs in the body because all the cells, the lymphocytes, are there to attack them. The phagocytes and the lymphocytes are there to destroy them. So they have no PALs. PAL means friend, right now. So no PAL. N for neutralization, O opsonization, P for precipitation, A for agglutination, and L for lysis. Easy to remember, right? Let us look at the first type of action. It's called neutralization. Now, in neutralization, the antibody called antitoxin binds with toxins which are produced by bacteria, and thus it forms the antigen-antibody complex. Now, in this way, it neutralizes the toxin. The antibody neutralizes the toxin. The second type of action is called opsonization. Here we have a bacterium with the antigens on the surface of the cell. So the antibody opsonin combines with the antigens, and then it acts as a marker so that the phagocytes here can easily recognize that this is a foreign particle, that this is an antigen, and thus they will move towards it and destroy it by phagocytosis, by ingesting it and then digesting it with enzymes. So basically, the antibody opsonin is a marker. It tags the antigens as foreign substances so that the phagocytes can easily recognize these as antigens, these bacteria, for example, and then it will destroy the bacteria. The phagocytes will destroy the bacteria through phagocytosis. In the third type of action, precipitation, the antibody called precipitin, shown here, reacts with dissolved antigens. So some antigens are soluble, and when they react with this antibody called precipitin, the precipitin will cause them to be joined together, to combine together, so that they form an aggregate and they form an insoluble complex. So there will be precipitation of antigen molecules out from the solution, and they'll form an insoluble complex that can be easily ingested by the phagocytes. The fourth type of action by antibodies is called agglutination. So in agglutination, the antibody called agglutinin coagulates and clumps the pathogens together and make them easy targets for the phagocytes to ingest and destroy. For example, here we see the antigens on the pathogens, the foreign particles on the pathogens, and this is the antibody called agglutinin. So agglutinin will bind to the antigens on the surface of the pathogens, and they will cause them to clump together, to combine together to form a mass of pathogens, so that these pathogens are not so mobile and they cannot move easily. Thus, they become easy targets for the phagocytes to ingest and destroy. So think of the word glue, is it agglutination or agglutinin? There's this word glue here. So it implies that these pathogens can be clumped together so that they can't move around freely. And then the phagocytes are able to move towards them and then ingest them with the pseudopodia and finally to digest them with the enzymes. The fifth action of the antibodies is called lysis. In lysis, the antibody lysine combines with antigens on the bacteria or virus or other pathogens, and this causes bacteria to be broken down and decomposed. For example, in this picture, we see a bacterium. On the surface of the bacterium, there are the antigens, which are recognized as the foreign particles by lymphocytes. So lymphocytes produce antibodies to destroy the bacterium and the antigen. So the antibody lysine combines with the antigen on the bacteria to form an antigen-antibody complex. Next, the bacterium will then undergo lysis, which is the breakdown and decomposition of the bacterial cell. So usually what happens is that the antibody lysine will cause the breakdown of the cell wall or the cell membrane. And this causes the rupture of the bacteria or the bursting of the bacterial cells. This is something like the analogy of a soldier that throws a bomb into the enemy's territory and causes an explosion. Here's a summary of the 5 actions of the antibodies. So firstly, there's binding of antibodies to antigens to form the antigen-antibody complexes. And this will inactivate antigens by neutralization through the coating of toxins with the antibody to neutralize the toxin, through agglutination of pathogens such as bacteria, where the pathogens are clumped together, and then through precipitation of dissolved antigens, so that the dissolved antigens will precipitate out as a solid that can be easily engulfed by the phagocytes. Through lysis of bacteria, where the bacteria is broken down, cell walls are broken down, and the bacteria will burst or rupture or get decomposed, become decomposed. And through opsonization of antigens, where the opsonins act as markers to mark the antigens as foreign substances. So all these actions will enhance or stimulate phagocytosis by the macrophages, which are the large phagocytes that ingest the antigens and then destroy them by using enzymes. Now for this lesson, there are no questions yet, but in the future, when I am free and have more time, I will put up some questions concerning antibodies and concerning the subtopic of 11.2 in the description box below. So that's all for this lesson, and in the next lesson we'll be discussing subtopic 11.3, types of immunity. Here's an important announcement for Form 5 students. There'll be an online biology intensive revision course for Form 5 students that will be carried out from November to mid-December 2020 to help those who are preparing for the SPM in January next year. So to all Form 5 students, during this period of MCO, if you have a lack of guidance on your revision for biology and you're wondering how to revise your biology, then you may be interested to join this intensive revision course and there are about 30 more places left, so if you're interested, please refer to the description box below to get the details. And you can email me to find out more about this intensive revision course. So thanks for watching and stay tuned for the next video. Please share, like and subscribe. Goodbye for now.